Welcome to class number 3, session number 2. During this session, we will learn about the main components of a sensor and different ways to classify the sensors. A sensor is a transducer that converts the measurement, namely a quantity or a parameter, into signals that carries the information. Chemical sensors usually contain two basic components connected in series. The first component is a chemical or molecular recognition, which we call usually a receptor, and the second part is a physiochemical transducer. In the majority of chemical sensors, the receptor interacts with the analyte, and as a result, the physical properties are changed in such a way that the appending transducer can gain an electrical signal. In some cases, one and the same physical object acts as a receptor and also as a transducer. Sensors can be analog to the human senses. In both cases, signals are provided from the environment and converted into a signal by a sensory receptors in humans or by receptor and transducer systems in sensors. In the amplification and the processing stage, the sensory nerve system is comparable to an analog circuit. The processing stage so far, which is obviously in the brain for humans, will be performed by a computer in the case of sensor system. A modern definition of the sensors includes several features. The first feature is that the sensor has to be in direct contact with the investigated subject. The second feature is that the sensor has to transform non-electric information to electric signals. The third feature is that the sensor has to respond quickly and also operate continuously, at least in repeated cycles. The other features is that the sensors has to be small and also inexpensive. The main components of sensors may include the following parts. The first part is a receptor. Receptor is an agent that is able to interact selectively with particular substances or mixture of substances, catalyze reactions selectively, or participate in chemical equilibrium together with the analyte. Typical for biosensors is that molecules are recognized by their size of dimension, namely by the so-called steric recognition. The second part of the chemical sensors is called transducer. This part is a device that transforms actual concentration value or non-electric quantity into an electric quantity, such as voltage, current, or resistance. There are several types of transducers, and the two main representative ones are the following. Energy conversion transducer, where the principle of the energy conversion means that the electrical energy is produced by the sensor. Many of these kinds of sensors are able to operate without any external supply of voltage. The second part of representative transducers is called resistive transducer. A transducer that transforms environmental properties to conductivity, or on the other hand, resistivity, of electrically conducting materials is a transducer that answers this property. Sensors or chemical sensors can be divided into various categories according to their operation mode. In the following slides, I will make a brief explanation on part of these categories, one by one. The first category of these sensors are the chemical sensors, namely devices that transform chemical information, like the concentration of a specific sample component, into an analytically useful signal. As mentioned in the previous slides, chemical sensors contain two basic fundamental units. a receptor, and a transducer. In the receptor part of a sensor, the chemical information is transformed into a form of energy which may be measured by a transducer. Based on that, electrochemical sensors are a type of a chemical sensor. Indeed, in continuation to the chemical sensor, I would define right now the second category of the sensors, which are the electrochemical sensors. These sensors are the largest group of chemical sensors. They can be used to detect the majority of common toxic gases in a wide variety of safety applications. Electrochemical sensors operate by reacting with the gas or analyte of interest and by producing an electrical signal that is proportional to the analyte concentration. A typical electrochemical sensor consists of a sensing electrode, or we call it usually working electrode, and on the other hand, a counter electrode that is separated by a thin layer of electrode. This electrolyte composition and the sensing electrode material is selected based on the chemical reactivity of the target. Electrochemical sensors are compact, require very little power. Also, they exhibit excellent linearity and repeatability, and generally have a long lifespan, typically 1 to 3 years. More details on these sensors will be learned in the upcoming classes. The third category of the sensors are the so-called biosensors, namely devices for the detection of an analyte that combines a biological component with a physicochemical detector component. As an example of biosensors, I present here on the slide the results from a research that is taken and obtained by a team of researchers with the United States Department of Energy at Berkeley, who has invented a technique in which DNA or RNA assays can be read and evaluated without the need to elaborate chemical labeling or sophisticated instrumentation. This biosensing approach is based on the electrostatic repulsion, in which objects with the same electrical charge repel one another. One of the most amazing things about the electrostatic detection method is that it requires nothing more than the naked eye to read out results that currently require chemical labeling and confocal laser scanning. The first category of sensors are the optical sensors. This category of sensors can be categorized into two main subgroups. The first subgroup is called proximity sensors, which are designed for use in detecting the presence of an object or motion detection in various industrial, mobile, electronic applications, and retail automations. Examples of proximity sensor usage include detecting an out-of-paper condition in a printer or a mobile phone screen that dims to save battery life when placed near a face. The second subcategory of the optical sensors is called ambient light sensors, which provide precise light detection for a wide range of ambient brightness and are commonly used in LCD backlight control in mobile phones, LCD TVs or panels, and notebook applications. One way to convert the optical signal is by using electro-optical sensors that are electronic detectors that convert light or a change in light into an electrical signal. Light have many components that can be sensed, like the wavelength, the intensity, the polarization, and the phase. The interaction of the light with the matter can be measured by means of absorbance, reflectance, luminescence, and more. More details about these sensors will be given in the next classes. The fifth category of the sensors is the so-called mass-sensitive sensors. These sensors can detect analyte molecules by sensing its mass. There are several kinds of mass-sensitive sensors. The first subcategory of mass-sensitive sensors is the cantilever sensors. In this subcategory, the displacement of the beam on a cantilever surface is related to the applied force and length of a beam. The surface of the cantilever can be modified, allowing a differential stress to propagate as an analyte of interest is bound to its target on the modified surface. The second subcategory of mass-sensitive sensors is the so-called quartz crystal microbalance, or briefly QCM. QCM is an ultra-sensitive mass sensor that relies on a quartz crystal that oscillates at its resonance frequency due to the piezoelectric effect. Upon addition or removal of small masses or molecules from the surface, the resonance of the quartz crystal is disrupted, thus leading to the change in the measured frequency. The prompt changes in the frequency are then transformed throughout a physical relationship to changes in mass. The third subcategory of mass-sensitive sensors is called surface acoustic wave, or briefly SAW. A surface acoustic wave is an acoustic wave traveling along the surface of a material exhibiting elasticity with amplitude that typically decays exponentially with the depth into the substrate. As the acoustic wave propagates throughout the surface of the material, any changes to the characteristics of the propagation path affect the velocity or the amplitude of the wave. Changes in velocity can be monitored by measuring the frequency or phase characteristics of the sensor and can then be correlated to the corresponding physical quantity that is being measured. In summary, there is a large variety of sensors, but the basic analysis and parameters that characterize the sensors are common to all sensors, as we will learn in the next session. This is now the end of class number 3, session number 2. Thank you.